So fermented foods are living foods.
Mm-hmm.
And anything that's alive is kind of unpredictable.
Yeah.
I think that's why through most of the industrialization of our food supply, chemistry was the answer, adding compounds. Maybe the past 10 years, we've turned a page to go back to say, what is the biology of the foods we eat? And so with that comes a lot of opportunity, but also the issue that microbes' life can be unpredictable.
Hello and welcome to this episode of the Pittcon podcast. Today we sit down with Maria and Marco to discuss how we can improve diets and reduce food waste with fermented foods. Please could you briefly introduce yourself and your current research focus?
Sure. So Maria Marco, I'm a professor in the Department of Food Science and Technology at the University of California, Davis. I'm a microbiologist, and I have to say I'm passionate about microbes that are good for us, good for our health, and particularly those we find in foods and our digestive tracts. Now, add on a little bit more, get a little more specific. I'm particularly interested in microbes that we call lactic acid bacteria. It's a fascinating group of microbes that are essential for making fermented foods, and we come across as being called probiotic. And so, yeah, I'm interested to know why, how, what do they do, how they're good for us.
And talking of fermented foods, what exactly is meant by a fermented food, and why have they been hitting the headlines so much in recent years?
So fermented foods are foods made by microbes. That's all you need to know. They're beverages as well. So wine, beer, maybe you've heard of kombucha or kefir. These are fermented beverages. And bread, sauerkraut, yogurt, chocolate, coffee, the list goes on. These are fermented foods. These are foods we wouldn't have if it wasn't for the microbes that make them. And your second question is why? Why fermented foods? I think there's two main reasons. So one of these reasons is that fermented foods are kind of in the headlines. It's a question. Is that chefs around the world at the top restaurants have started making fermented foods, exploring fermented foods because of the diverse flavors, the sensory properties that they give. And so that's one reason I think fermented foods have become more popular. Another is that dietitians, experts in nutrition have started to look to fermented foods because of the health benefits they could bring. And at least in some circles, like say in the US, they're ranked as the number one superfood by dietitians for like 6 years running, above salmon, above berries. So that's pretty interesting. I don't think we have all the answers to why or if this is entirely true, but certainly the belief is out there that fermented foods are good for us.
Do you think that belief has helped to kind of catalyze interest in research in a way? Like, do you think it's been beneficial to your research community?
Yeah, 100%. So much of the research we do is driven by public interest, what's seen to be important and valuable. Of course, fermented foods have been made for thousands of years, so they're not new. But what we can bring now with the tools we have available, what we can do in the lab is so much greater than we could do 10, 20 years ago. So it is a really unique moment to bring together the basic understanding of fermented foods to the bigger picture of how they can help our health.
And kind of building on that and thinking about how your research can be beneficial to the public, what impact can the study of fermented foods have on matters of safety, diet, and even food waste?
Yeah, thanks for that question. For those three areas, I can break this down. So for safety, Fermented foods are generally safer than the ingredients used to make them. And for most of the world, unfortunately, where access to clean water is still a problem, drinking beer could be safer than drinking the water. And that's true, really, for much of human history. Of course, when you do have access to clean water, that's not really the safety issue we're talking about. But they are generally safe because of the chemical environment that maybe have an acid flavor that's creating an environment that pathogenic microbes don't like.
Yeah.
So the second point was on waste. Let's talk about waste. So we can use fermentation. We can take foods that are going out, so spoiled or a little bit defective, and upcycle them through fermentation. We can take those lactic acid bacteria that I mentioned and ask them to ferment imperfect fruits and vegetables and make them, into a new product or desirable again. When we ferment, we're preserving food. So the berries, the cabbage, they'll all last much longer. And I forget your third. The other one. Diet. So why in the diet? Yeah, that's really the crux of the matter. And we think about the excitement over fermented foods today. What's pretty fascinating is that, if we have a cabbage or we have a cucumber, we know generally that's healthy. Yeah. And what happens when we add a little salt and let those microbes grow? They're digesting the food for us, so they're making it more easily digested by us when we eat it. But those microbes are also giving new bioactive compounds to these foods. So I really think what we're seeing is that, these microbes are changing milk, berries, cabbage, etc., into something that is more nutritious than what we started with.
That's fascinating. So in your talk, or your Pittcon talk, you mentioned the unique challenges in the production of fermented foods. Could you elaborate on these challenges, especially in relation to microbial growth and metabolism?
Yeah, thanks for the question. So fermented foods are living foods. And anything that's alive is kind of unpredictable.
Yeah.
I think that's why through most of the industrialization of our food supply, chemistry was the answer, adding compounds. Maybe the past 10 years, we've turned a page to go back to say, what is the biology of the foods we eat? And so with that comes a lot of opportunity, but also the issue that microbes' life can be unpredictable. So just as we need microbes to make fermented foods, some of those same microbes, or sometimes unwanted contaminants, contributors, make the foods spoil or have quality defects in how they look or how they taste. So when we're making fermented foods, we're constantly managing our microbial gardens that we don't want weeds in there. And so I think that's a particular challenge with fermented foods.
I really like that, the metaphor you use of a microbial garden, because I think it really illustrates the diversity of microbes that we have and how there are just so many different types out there, and they all kind of help and create and make something, that's useful and very, good for us in a way.
Yeah, absolutely. Just as we have a garden with maybe a row of carrots, each one of those carrots may grow a little bit differently and have maybe slightly different, of course, look to them and even flavors. So we could say that would be just one species of microbes and all the variation there. But certainly if you want to have a nice salad, you need to put different vegetables on there and different fruits and vegetables. So I think that's what we're looking at with fermented foods.
No, definitely. And looking specifically to one industry, how do industrial starter cultures in cheese production in particular impact manufacturers' reliability and consistency?
Industrial starter cultures are microbes that are grown specifically in pure culture, usually, or well-defined, and have transformed making fermented foods from small scale to a very large industrial scale. So we wouldn't have the beer, the wine, the cheese that we have today in such scale if it weren't for starter cultures. They work most of the time, but sometimes they don't work as planned. And so even in the best scenario, microbes will be microbes, and it's not completely unpredictable. You can't control it all the time. So starter cultures are really important. They've been really valuable for the industry, but they're not infallible. And where we don't have starter cultures right now will be making our fermented fruits and vegetables. We have starter cultures for making yogurt. We have starter cultures for cheese. So I could see an area of growth in making fermented fruits and vegetables more accessible is to have starter cultures available that are making those foods more reliably made with fewer defects.
Is that something that you think is a problem in industry, or is it, say, like a funding problem, or maybe even we just simply don't really know what the best approach is to do that?
I think a little bit of both. Yeah, scaling up the growth of these microbes and the sort of ecology that happens in these particular fermented foods can be a little bit more complex. And so I don't think it's an insurmountable issue. I think it's a kind of industry will and the basic science needed to get there.
Hopefully one day. So speaking specifically to the different technologies and methodologies that you use in this area, what techniques are currently used to measure and monitor the quality of fermented foods? And how have these methods evolved over time?
So like much of microbiology, or from medical to environmental, we still rely on 19th century methods.
Right.
So culturing the bacteria, as you would imagine, like a microbiologist to do on a Petri dish in the lab, or maybe a lab coat on and goggles. I don't know, but that's still the most common way. But it's problematic. As I said, it's a method developed in the 1800s. And so what we've turned to is to use more culture-independent methods where we don't grow those bacteria on a Petri dish, but instead look at their nucleic acids, their genetic material, to inform us of who they are and what they're doing. And there's a lot of power in that because we can look at whole communities of thousands of different microbes with hundreds or thousands of different species all at once. And so it is a very powerful technique.
That leads brilliantly onto my next question. So in your presentation, you specifically mentioned the use of culture-independent microbial analyses and DNA sequencing. If you could maybe give some case studies of how have you seen these techniques transform the way that we identify and diagnose product defects in fermented foods?
Yeah, actually, we've been pretty successful in using them, and sometimes I'm even skeptical how well it'll work. But we were able to sort of identify, would be a smoking gun of spoilage that I was convinced wasn't microbial, but we went back and through sequencing, through DNA sequence analysis, looking at the microbial composition on fermented olives. We did the same thing with cheese to identify the most likely culprits. So for fermented olives, there was a spoilage event where the olives smelled and even tasted like they should. The flavors, but they were very mushy. And so we hypothesized it was some microbe that could eat pectin. So we identified the microbes in the olives and found through culture-based methods as well that pectinolytic yeast are a problem on these olives, that you want to have a little bit of the softening. If you ever have an olive that can be really hard, especially you don't want to eat an unfermented or cured olive. But they should be a little soft, but not mushy. And so that was sort of the first step we took in looking at spoilage issues with fermented foods. And since we've applied similar principles to understanding how cheddar cheese gets slits or these cracks we were able to use the DNA sequencing techniques to reveal the bacteria that cause slit defects. So slits may be nice or endearing to us if we look at a cheddar cheese.
I was just thinking, I always think like, oh, it's a good cheese. It's it's crumbly. It's
crumbly. But yes. But when you're making a lot of cheese and the purpose of that cheese is to slice it or grate it in the large scale production that this is, you can't have that. They'll have to throw away a lot of cheese when there's a slit. So we helped a company identify which microbes cause this as they would call it, a defect.
Wow, it's so fascinating. I think for me personally, and I'm sure others have the same idea, that when we think of spoilage, we think of mold. Whereas like, even texture, I never would have considered texture to be a sign of spoilage or the slits. Right. Like it's all these very kind of almost visual characteristics or even textural characteristics. It's fascinating, really.
One person's treasure is another person's trash.
It's true. It's very true. It's very fascinating, but very true. Mm-hmm. So, moving on to the next question. Looking specifically to strain-specific diversity, how can exploring this contribute to the development of new fermentation processes, particularly for fruit and vegetables?
Yeah, it's a really fascinating question, and just as we are people, maybe with different interests and different favorite fermented foods or what we like to do in our spare time. If I take two isolates of our favorite bacterial species, Lactic, Bacillus plantarum, very long name. Anyway, it's a species, and we take two, even from the same food, they can have completely different properties in the sense that one does a really good job at fermenting the food and the other does not. But yet they coexist together. So we're getting into really fine scale and a deep resolution of understanding when we get down to strain specific differences. Although they're the same species and they'll still make lactic acid because they're lactic acid bacteria, we would have really different outcomes in a fermentation if we put this one that does well and one that does not. And so we're interested to understand how these two very different members of the family can live together and what happens when we take them apart, and can we apply one that does well to kind of boost the fermentation, and what do we lose in that fermentation when we remove the other one? I mean, it seems to be there for a reason. Maybe it's there to help its friends, even though it isn't the best grower. So we're reaching the edges of what we can do with DNA sequencing type methods. When we go to that strain level differences, we can look at the genomes. We know they're different genetically, but we still have a lot of questions of what that means and picking them out from each other when we use DNA-based approaches in the community of the fermentation.
And do you find that there's quite a large genetic difference between the two? Like how, in your experience, like have you noticed that the similarities do confer to different, say, like genotypes or fermentation boosting properties?
So, and their core genome would be very much the same. They are the same species. There's no doubt about it. But we call it the accessory genome, where these bacteria have acquired different traits, maybe to make polysaccharide, for example. Polysaccharide can make fermented food slimy.
Oh, wow.
Right? So maybe you do want that, but you're right, you don't have a choice if you don't know what you're putting in there or you don't know what's in there. So maybe it's making a bioactive compound that we want to deliver to the digestive tract in a fermented food. So you have to kind of carefully curate the microbes you're selecting to put into a fermented food. And we're not there yet. I mean, most of what we talk about industrial starter cultures, they're selected because they grow quickly in that food. They deliver the fermented food, but they're not selected because of the health benefits they can bring as microbes.
Do you think maybe one day there will be that push towards the health benefits? Like, I imagine there would be with the...
We could do it today.
Yeah. Go out there and tell them.
Yeah. No, I mean, I pushed it a little bit, but there's quite a lot of understanding out there. If we look at probiotics, there's certain core benefits we would expect, and where the edges of the science are, and understanding what are their specific enzymatic contributions or metabolic contributions they provide to us.
Mm-hmm. It's so interesting. Based on your experiences at Pittcon and also your research, building on from I suppose the push towards more of the health benefits, what other future directions do you see for the field of fermented foods research and drinks research?
Well, I really like the idea of this guided fermented food production where you're guiding for a specific health benefit. Frankly, we don't have a lot of human study evidence to rely on. We have the feeling, we may have a gut feeling that fermented foods are good for us, but there aren't many studies that verify this collectively for fermented foods or for individual fermented foods. So I would say we need to keep pushing to continue the study of these foods and ultimately make them accessible. It should be that we're not paying $10 for a little shot of sauerkraut or something. It should be something we can still make at home and available to everybody.
With so many headlines focusing on the health aspects of it and not so many focusing on the food waste side of things, like you spoke about, how we can take something that's close to being spoiled but then make it and evolve it something new. Why do you think that is? Why do you think there's so much focus on what's trendy and like what is that gut feeling good for us, even though there hasn't been a lot of research, whereas there are some very concrete studies that do show the benefits on food waste and sustainability. Why do you think there is that difference?
Yeah, I think it comes down to economics and what is, when it becomes too expensive to put food into a landfill or we start regulating against that, then there's the economic need or the push to protect our food supplies. And if we can get those fermentations a bit more under control, then really there is no limit. I think you have to a little bit rebrand the food, but so long as it tastes good, people are going to want to eat it. Right? If it doesn't taste good, it's not going to last very long on our supermarket shelves. So, I think there's challenges along the complexities of the food processing and distribution lines. Like, how do we get the food from the supermarket or from the farmer and then actually use it? So I think there's some logistical challenges to work out. And maybe that's why we don't hear about this option as frequently as we do about the benefits of these foods.
Pittcon Perspectives, unforgettable Pittcon moments from the experts.
So, as we mark the 75th anniversary of Pittcon, could you share your first memory or experience of attending this conference and how it's impacted your view on your scientific community and the scientific community as a whole?
This is a really vibrant conference, and what strikes me about this meeting is the diversity of disciplines represented. I feel like it's a big tent for chemists, biologists, physicists, mathematicians, potentially anybody who has an interest in understanding the basic chemistry of life and the planet, frankly.
Mm-hmm. And what are you most looking forward to at Pittcon San Diego this year?
Got to go to the expo. The product show is where it's at. So, but yeah, it was the scientific sessions. It's difficult to know where to go. There's so many exciting events happening at once. So these are full days.